Let us learn about the rDNA genes. Now, whenever we speak about rDNA, the first thing that should come into our mind is the nucleus, because rDNA and most of the components of rDNA are found in the nucleus. So if you look at the cell right here, you can see that this is the cytoplasm and this is the nucleus, and right there is a small portion known as nucleolus. Now, nucleolus is quite different from the surrounding material that is found around it. That's basically the nucleus, because the nucleolus is related to rDNA more specifically. So now let us look exactly where in the nucleus and how in the nucleus the rDNA is found. So if you zoom in and look into the nucleolus, you can see that there are clusters of DNA here, and then but how are these formed? They are formed in a cell by rDNA, rRNA, ribosomes, and rRNA polymerase. That's basically RNA polymerase 1. And all of these components that are found only specifically in the nucleolus lead to the formation of clusters of rDNA genes. Now, let us look at the mechanism in which there is a formation of the basic raw material that is required for the formation of the rDNA. And in order to obtain this rDNA, we need the transcript. So basically, there's the DNA. We know that it goes from the DNA, and then we have a strand of DNA or a messenger RNA. And then the messenger RNA, it gets transcribed. That's basically the transcribed RNA, and then the transcribed RNA gets replicated, and then there's a formation of the DNA. And this entire process is also seen in the formation of a completely new rDNA. That is taking a strand of the rDNA, a pre-existing rDNA, and getting a transcript out of it. Now let us look at the mechanism. How does this happen? So that is the rDNA gene. And rDNA has three components, three units into it that acts as genes that give rise to the transcript. So we have the 18S, and we have the 5.8S, and then we have the 28S. And all of them together give rise to the pre-rRNA or the RNA transcript. This is also known as the 45S RNA. Now, this pre-rRNA, it's a pre-rRNA, so it undergoes further maturation, and maturation is promoted by endonucleation and also by the modification of the bases that are found in the RNA. Modification and endonucleation leads to the formation of RNA subunits, the pre-rRNA subunits, namely the 18S subunit, the 5.S subunit, 5.8S subunit, and then we have the 28S subunit. But the process does not really stop here. Let us now go back and look at the cell. So the location of the subunits is inside the nucleolus. The maturation is further, there's a need of more further maturation, which does not really takes place inside the nucleolus, but the entire transcripts, all of these three transcripts, subunits of the transcript have to travel outside the nucleolus. So they travel from the nucleolus into the nucleus, and then eventually from the nucleus until the cytoplasm. And once inside the cytoplasm, these are known as the 43S complex. All right, so now let us observe the mechanism and the transcript. Okay, so let us observe the mechanism and how there is a transcription of the rDNA gene. Now, if you look at this diagram right here, you can see that the rDNA consists of a promoter. So the first step is the coming in of the RNA polymerase 1. RNA polymerase 1 brings with it two complexes, the UBF1 and then there is an the SL1. So two transcription factors two transcription factors plus transcription factor 1 and transcription factor 2. The first transcription factor, namely, is known as UBF, and the other is known as SL1. Both of them accompany RNA polymerase 1 on the promoter region of the DNA. Now let us look at the transcription factor in more detail. UBF1 is involved in a lot of things. It binds to the upstream direction of the DNA. It influences the activity of the polymerase. It regulates transcription and decondenses chromatin. Now, the second transcription factor is also known as SL1. SL1, structurally speaking, is a quite complex factor. There's a TATA binding protein that is found near the one end. Then we have three different types of TATA associating factors, namely 48, 68, 110/95. And then recently there was also a discovery of two additional TAFs, namely TAF41 and TAF12. Now, the question is, what is actually the function of SL1 and UBF1? Why are they here? So what they do is that they both align the entire RNA polymerase. One. Now let's take a look at the structure of the rDNA molecule. An rDNA, a typical rDNA is quite big. The length of a typical rDNA is about 45 kilobase pair long, and it is mostly situated on chromosome 13, 14, 15, 22, and 21. Now, there are a lot of parts of this entire DNA that have different functions. So now let us take a look at all of these components and parts of the rDNA. The one that we're looking here are known as coding sequences. Coding sequences are transcriptionally active and are about 13 to 14 kilobase pair in length. And the spaces that are in between them are much more larger. They are known as intergenic sequences and range about 30 kilobase pairs. Then we have the promoter region. The promoter region is usually at the upstream direction, that is at the 5' end. And it also consists of a lot of components. So the promoter region consists of the promoter, of course, then the promoter spacer, and then we have repetitive enhances. And these are repetitive nucleotide sequences. Moving forward, there's also at the end, you can see that the dark purple region right there is known as the terminator region. So let us look more into details about the components, about all of the components of this entire DNA molecule in order to understand it more better. So the first component that we're going to be looking into is known as the promoter region. If we isolate it and look at the structure of the promoter region, we can notice that it is made up of a lot of parts. So it has a start site, and then adjacent to the start site, we have a core element. Adjacent to the core element is the UCE, or the upstream control element, which is approximately 100 base pair upstream. As you can see, there is an invisible divide between two major portions of this of the promoter region, which means that this region is bipartite. That means it has two parts. So that was all about the promoter. Let us look into another component of the rDNA. So this is known as, let us look at this component right here. This is one of the core elements. If you look closer here on this structure, you can see that it's also made up of two parts that flank the upstream region, the 5' region, and the 3' region. And these structures are known as the terminator elements. So two terminal elements on both sides. And due to the fact that there is an upstream region and a downstream region, there are elements that are named according to them, and their presence is according to the upstream and the downstream region. So we have the T1 to T10 that are found in the terminator element, and then we have the T0 that are found in the upstream element. Then lastly, we have the TSB that is found near the enhancer. Let us initiate transcription. Transcription is initiated at the T0 region on the upstream direction of the rDNA. So where is this T0 region now? We know for the fact that the upstream is in this direction, approximately at the, reaching the 5' direction of the DNA is the upstream direction. But then where exactly is the location of the T0? The T naught or the T0 is located at the is also known as a terminator site that is located in the upstream direction and is a major point of the initiation of transcription. If you look at this position, this location more closely, you can see that it looks somewhat like this. And of course, there are the two components, the TTF1 and the CSB, that initiate the transcription. But then, along with TTF1 and CSB, there are another components that are involved. So there is the RNA polymerase 1, and then there is a transcription factor 11H. So both of them, plus the TTF1 and CSB, initiate transcription by binding together all together and attaching themselves to the T0 site. So let us look at another component that is involved with rDNA, known as the TTF terminator. Now, what is the TTF terminator? The TTF terminator is quite important in initiating termination. That is, it binds to the region where the protein needs to be truncated so that there is the release of the 45S DNA or the 45S RNA, and also is involved in epigenetic regulation. But the function of the TTF terminator depends according to where it is actually it binds. So if it binds near the initiator part, near the coding sequence of the initiator part, to be precise, the T0 terminator, in the upstream direction, it can initiate transcription. However, if it binds on the other side, known as the T1 to T0 terminator, which is present downstream, it leads to the release of the 45S strand of RNA. Now, the main question is, let us look at how is there an initiation of transcription with the help of the TTF. For the initiation of transcription, the TTF does not like to work alone. It works in conjunction of another entity, of another element known as the CSB. So CSB is also known as the B protein for the cocaine syndrome. If there is no CSB or the B protein for cocaine syndrome, there can be a pathology known as cocaine syndrome, where the individual is very sensitive to light. Now, the job of these both is to initiate transcription. And in order to initiate transcription, they need to follow a series of steps. So the step one is to remodel chromatin. We know that chromatin is packed inside the nucleus. So we need to loosen this chromatin up by taking care of the DNA and breaking the DNA interactions. Now, how can this be done? So the taking care of the histones is done with the help of a histone acetyltransferase or histone methyltransferase. And here especially the case is with G92A. So G92A is a type of histone methyltransferase that helps in the loosening of the histone protein with the attack on the lysine 9 of histone number 3 and the Y subunit of histone protein 1. And in this entire process, there's a consumption of ATP. So we know that in the core of the nucleosome, there are 4 histone proteins. So there's a special attack on histone 3 and then histone protein 1, which is the linker, especially the Y subunit and the lysine of 9. We know that there's always always an attack on lysine residues that are present on the tails. We learned about this earlier. So that's how there is the breaking of the histone, the attack on the histone, which leads to the loosening of the chromatin. Now, however, the job is not over. There's a need for another element that helps in the maintenance of the entire thing. And the maintenance is done with the help of MB protein, and especially the MBD3 domain of the MB protein. Now, what MB protein does is that it goes and it attaches itself to the parts of the chromatin that are not methylated properly, or the D or the unmethylated region, and keeps this region in a hypomethylated state so that, maybe they don't interfere with the entire reaction. And last but not the least, after this entire process is over, there's another protein known as the GADD45A that helps in the maintenance of this entire structure. Then there's an initiation of transcription.